All right, welcome back to Biochemistry on Catalyst University. My name is Kevin Tokoph. Make sure to like this video and subscribe for future videos and notifications. In this video, we're going to talk about heme degradation, initially to bilirubin and then the further degradation products that occur in the large intestine in humans. Now, we have two main sources of heme for degradation. The first is obvious, the heme that's attached to hemoglobin, which is on your red blood cells. That's the heme that carries oxygen in the blood. And as we hopefully know at this point, after a certain amount of time, usually around 120 days, the red blood cell, its lifespan is over and it gets degraded. Now, if the red blood cell gets degraded, the hemoglobin and the heme itself also have to be degraded. Okay, so that's one of the sources of the heme. The other source is from the diet. So we have to get iron in the diet. It's an essential mineral. And so one of the sources that iron is heme. And so when the heme is initially absorbed, in order to free the iron, the heme is actually degraded to release the iron, and the iron gets initially stored in ferritin. Okay, so those are the two main sources of heme, and they're going to follow this pathway. All right, so as you can see right here, this is heme. You can see the iron in the center. That heme, if it is initially attached to a red blood cell, it will be removed from the red blood cell and oxidized into biliverdin by the enzyme Heme Oxygenase. This enzyme is also obviously in the intestinal cells. Once heme is absorbed, Heme Oxygenase can react with heme, oxidize it into biliverdin. Okay. Now, hopefully, what you notice is that this opens the ring at this point designated alpha up here. It opens the ring, and we end up with biliverdin. Okay. Now, biliverdin can be further reduced into bilirubin. This is catalyzed by biliverdin reductase. The reducing equivalents come from NADPH, and we get bilirubin. All right. Bilirubin right here is sort of an important molecule in the crossroads of heme degradation because it poses a problem in newborn infants that have jaundice, which we'll get to in just a minute. But to understand bilirubin, take a look at this picture up here. Bilirubin, and I'll go ahead and show you this, it's normal isomer, because notice it has some double bonds right here, number 1, and number 2 right here. These are the important double bonds. Those are chiral double bonds, and normally the normal isomer that's produced from biliverdin reductase is the ZZ isomer. Okay? In the ZZ isomer form, this bilirubin molecule has internal hydrogen bonding, which basically prevents these potential hydrogen bonding groups from interacting with water, which makes this molecule of bilirubin insoluble. Okay, it's not soluble. So what the liver macrophages have to do is they have to conjugate bilirubin to glucuronide residues. You can see these carboxyls up here get conjugated to glucuronide residues. That's catalyzed by a specific glucuronyl transferase, and that is UGT1A1. All right, and that makes bilirubin diglucuronide. All right, these three reactions, if we're talking about red blood cell degradation, that is, heme oxygenase, biliverdin reductase, and this glucuronyl transferase, these occur in the liver macrophages. Okay, once you form bilirubin diglucuronide, it gets exported to the intestine, okay, for potential excretion, all right? And we're going to go over this whole thing in just a minute. But basically, by conjugating bilirubin to these glucuronide residues, these are polar molecules, glucuronides, and they solubilize the bilirubin so that it can be exported through the blood into the intestines, okay? So hopefully that makes a little bit of sense. Now, in some individuals, some newborns who have jaundice, they lack this enzyme initially. They end up expressing it eventually, but when they're born, for whatever reason, there's not enough of this enzyme, this glucuronyl transferase. So they cannot solubilize the bilirubin. And so the bilirubin just stays there in its insoluble form. And the problem with that is that if bilirubin accumulates to any extent, it causes all sorts of neurological problems that ultimately result in retardation and potentially death. So you don't want a lot of this around. So what they figured out is, and like I said, this bilirubin, its normal isomer is this ZZ. All right, so what can happen is this molecule, you shine UV light on the infant. Okay, it's not super powerful, but it's UV light. And what that does is causes a photoisomerization of ZZ bilirubin into ZE. Now, one thing that's important to understand here is the ZZ isomer, which is this one right here, has the potential for internal hydrogen bonding. Okay? Once that photoisomerization occurs into the ZE isomer, that internal hydrogen bonding does not occur. And so those hydrogen bond donors can now interact with the watery medium around it and makes it soluble so that this can be excreted into the bile. Okay? Additionally, there's also another one we don't really talk about. This bilirubin ZZ, if you expose it to UV light, it will actually undergo a cyclization reaction, which can also be excreted into the bile and urine. All right. In fact, this is actually a graph right here, a chromatogram, that shows exactly what we're talking about. What we say is that the bilirubin, that's the ZZ, the normal one formed by biliverdin reductase, okay, that cannot be excreted, okay, it's very slow to excrete, and as a result of that, it builds up. So this is the natural isomer. This peak is the natural isomer, the ZZ. See how long it takes to excrete, whereas if we expose it to light and isomerize it into the ZE form, notice that the peak for the ZE occurs much sooner than the other one. So we can see here the chromatogram that we export this one and excrete it a lot quicker than the ZZ isomer. Okay? And so by excreting it, it prevents the problems associated with newborn jaundice because they lack this enzyme that solubilizes the bilirubin into its diglucuronide form. Okay, now we have this bilirubin diglucuronide. What happens to it? Well, as we can see, it gets exported into the intestine. And in the intestine, we know that we have bacteria. Specifically, the large intestine has the highest concentration of bacteria. So what the bacteria do is they take this bilirubin diglucuronide and they deconjugate it. What deconjugation means is they remove these glucuronide residues so we get right back to bilirubin. Okay, so initially in the liver we have conjugated bilirubin. It's excreted in the bile, through the bile duct into the intestine. And the bacteria, the microflora in the large intestine specifically deconjugate bilirubin diglucuronide into bilirubin. All right. Now, specifically in the intestines, there's a lot of bacteria that do this, but the most notable one are the ones of the Clostridium genera. Okay, these are bacteria that can transform bilirubin into a variety of other tetrapyryl degradation products. Okay, and one very important one is urobilinogen. In fact, we're going to see that not the immediate product, but bilirubin does get converted into urobilinogen, which gets converted into all the other degradation products. So let's take a look at what happens. So after the bilirubin is deconjugated by intestinal bacteria, here's the bilirubin, it can undergo several different reactions. So most notably, we're going down here. So you can see these are plus H2, plus H2, plus. Going down, these plus H2, those are reductions. And going to the right, where you see minus H2, that's an oxidation. So through a series of reductions, bilirubin gets reduced to urobilinogen. Okay? Urobilinogen can go one of two directions. It can either go down here through reductions to stercobilinogen, or urobilinogen can be converted into urobilin by an oxidation. Okay, so let's suppose it does the oxidation. Urobilinogen gets oxidized into urobilin, and as we'll see, urobilin is actually what gives urine its characteristic yellow color. Okay, urobilinogen can also be reduced a couple of times into stercobilinogen, and then stercobilinogen can be oxidized into stercobilin. In fact, stercobilin is what gives feces their characteristic brown color. Okay, so all of these reactions right here, all of these reactions are catalyzed by enzymes that are made by intestinal bacteria. So these are all microfloral bacterial reactions that are doing this. Okay, now we can make the bilirubin, but from then on, all of these other products, the most notable ones in green, are made through bacterial reactions in the large intestine. Okay.